Hello, good morning. Thank you so much for joining me for today's performance, From Chemical Engineer to Cancer Warrior. How many people here know someone who has cancer? Many of us do. In fact, approximately 4 out of every 10 Americans will receive a cancer diagnosis in their lifetime. But every day, science brings us closer to better treatments and hopefully one day a cure. This is the inspirational story of Dr. Jessica Winter, a chemical engineer from The Ohio State University, and the personal journey that enhanced her life and her work. From university professor and researcher to cancer warrior and entrepreneur, Jessica is determined to make a difference for cancer patients. Jessica is one of the pioneers in the new and growing field of nanomedicine, engineering tiny particles that can attach to living cells to help fight diseases. And the tiny particles that Jessica engineers are called quantum dots. Now, this model of a quantum dot may look pretty big, but in reality, quantum dots are tiny crystals made up of just hundreds of atoms. Now, if that doesn't sound small, consider this. You could fit billions of quantum dots into the little period at the end of this sentence. And because quantum dots are so small, they have some really amazing properties. For example, if you shine a special kind of light called ultraviolet or UV light on them, they glow or emit light. And you can change the color of that light just by changing the size of the quantum dot crystal. So bigger quantum dots will emit red light, and smaller quantum dots will emit blue light. And I actually have a batch of real quantum dots here with me that I'd like to show you. And these are encased in kind of a hard material. So if you look at them just as they are now, does anyone see any bright colors coming out of these quantum dots? No, not really. Do they look like what you see up there? No, they just kind of yellow or clear. Well, now watch what happens when I turn down the stage lights and I illuminate them with a UV light. Pretty cool, huh? So this UV light is energizing the quantum dots, and they release that energy as light. And the color of the light depends on the size of the quantum dot. Well, Jessica was making these quantum dots to use for biological imaging, so to light up structures like these nerve cells so that scientists can study them. But Jessica was also curious to find out if she could use her glowing quantum dots to help fight a big health problem, like cancer. So she started talking with doctors and pathologists at the James Cancer Center at The Ohio State University. And these doctors told her that one of the biggest challenges that they face in treating cancer is actually figuring out what type of cancer their patients have. And so to understand why this problem of diagnosing cancer was such a big issue to these doctors, we need to take a step back and understand what is cancer. And it may come as a surprise to some people that cancer is not just one single disease that can show up anywhere in the body. It's actually a group of hundreds of related diseases that all share the same underlying symptom: uncontrolled cell division or cell growth. Now, we know that cancer can show up in lots of different parts of the body: lung, liver, breast, brain, to name just a few. But diagnosing cancer is far more complicated than just knowing which part of the body is affected. And that's because no two cancers are alike. Let's look at breast cancer, for example. There are many types of breast cancer. So that means this patient's breast cancer can be different from this patient's, which is different from this patient's, and so on. And because each patient is battling a unique illness, not all cancer treatments will work the same way for every individual. But how can doctors tell one type of breast cancer from another? Well, it's hard, because different types of cancer cells look really similar. But cancer cells do have telltale molecules and proteins called cancer markers. They're just way too small to be seen under the microscope. To illustrate this, let's imagine that these blue circles represent 3 different types of cancer cells. And we can't tell them apart under the microscope. let's also imagine that those little telltale molecules and proteins, those cancer markers, are as visible as these big orange shapes. Now, if we could detect these cancer markers, then they would tell us what specific type of cancer cells we're dealing with. And doctors can use those to design the most effective treatments for their patients. Let's look at breast cancer again as an example. So there are 3 important cancer markers in breast cancer: the ER and PR markers, which are often associated, and the HER2 marker. If none of these cancer markers are present, it's known as triple negative. And identifying these different types of breast cancer is really important because they need to be treated differently. ER and PR breast cancers are treated with a hormone therapy. That treatment won't work for HER2 breast cancer. But a type of targeted chemotherapy can be effective. For triple negative breast cancer, general chemotherapy is applied. So identifying these different cancer markers allows doctors to use the most effective treatments and avoid ones that won't work. And this type of individualized diagnosis and treatment is known as personalized medicine. This is just a simple example. It's actually much more complicated because one patient can have more than one type of marker in their cancer cells. So that means that one patient may have none of the markers or just one, or they can have a combination of different markers in their cancer cells, and even that might affect how well a treatment works. So knowing the type and amount of these cancer markers is really important, and not just for breast cancer. Identifying cancer markers is important in lots of different types of cancer. But right now, the only way that doctors have to identify these cancer markers is not all that efficient. In order to identify these cancer markers, here's what doctors do. They start by taking a biopsy, or a small slice of the patient's tumor. Then they put that on a microscope slide, and they stain it with a brown stain that will stick to just one specific cancer marker. Then they look at it under the microscope, and this is what they see. Wherever that cancer marker is present, it will show up as brown. But remember, one patient can have lots of different types of cancer markers in their cells. So to test for multiple markers, doctors would need to take a bigger biopsy and prepare a whole bunch of microscope slides because they use the same brown stain to test for each marker. So if doctors wanted to look at these 2 cancer markers at the same time, it would be practically impossible to tell them apart. This is why the doctors that Jessica teamed up with were really looking for a better, faster way to diagnose these cancer markers. Well, Jessica had an idea. She had her multicolored quantum dots. What if she could target those different colored quantum dots to stick to specific cancer markers and light them up so doctors could see them and they could tell them apart all at once? So instead of looking at that one brown stain over and over again, doctors could tell the proportion of different types of cancer cells in a biopsy all at the same time. And now to really show you how this works, imagine that I'm a pathologist, and I am analyzing a biopsy sample from a cancer patient. So here's my biopsy, and the circles you see here represent cancer cells. And now these little orange shapes represent cancer markers that are in this biopsy. But these markers are way too small for me to see. And in order to design the most effective treatment for my patient, I need to find them. So I'm going to use my brown stain to find these different cancer markers in this biopsy. So my brown stain will only stick to one cancer marker at a time. So first, I'll prepare my stain to stick to the marker I have represented by a triangle. So here I go. I'm going to put on my brown stain. Okay, so now I've identified where the triangle cancer marker is in this biopsy. But there are more markers that I need to test for. So I'm going to need to prepare a new biopsy sample. And I will need to prepare my brown stain, again, this time to stick to, let's say, the marker represented by a circle. So here I go. Okay, so now I've identified the circle cancer marker in this biopsy. And I'll need to repeat this process until I've identified all the cancer markers. So you can see how this might become tedious and slow. Well, now I am going to need a volunteer from the audience to come up and be a scientist from Jessica Winter's lab. And this scientist is going to help me figure out a better way to diagnose this patient. Would anyone like to come up and be a scientist today? Come on up. Okay. Hello, what's your name?
John Henry.
John? John Henry. Okay, I'd like to introduce everyone to Dr. John Henry. He is a scientist in the lab of Dr. Jessica Winter. Okay. All right, so Dr. John Henry, and I need you to help me figure out a better way to diagnose this patient so we can get them the best treatment in the shortest amount of time. So you and Jessica have engineered these different colored quantum dots to stick to one specific cancer marker in this biopsy. Let's see what we find when we use Jessica's technique. Go ahead. Great job. Can I assist you, Dr. John Henry?
Yeah.
Thank you. Now I can see where these different cancer markers are and which ones we have in this biopsy all at the same time. So you've helped me get a speedy diagnosis for this patient so that we can get them the best treatment in the shortest amount of time. Let's give Dr. John Henry a round of applause. Very nice, you can have a seat. Thank you so much for your help. So now let's see how Jessica actually makes this happen. Now, the quantum dots that Jessica uses typically live in an oily solution. And as we know, oil and water don't mix very well. But Jessica wants to use these quantum dots on a biopsy from a human. But are our bodies mostly oil or mostly water? Water, right. So this is a problem. But Jessica is an engineer. So she was able to figure out a way to get those oily quantum dots into a water-based biopsy. She decided to put them in a capsule. And it's a special molecular capsule called a micelle. And these micelles are made up of molecules that have two very different ends. One end, pictured in pink, really likes oil, but it does not like water. So it's drawn to those oily quantum dots. The other end, in yellow, does not like oil, but it likes water, so it likes to move away from the quantum dots. And this allows these micelle molecules to spontaneously form themselves into a protective capsule around the quantum dots that will allow the quantum dots to dissolve into a water-based biopsy. And not only that, but Jessica can pack a lot of quantum dots into these micelle capsules, so they can be really bright and doctors can clearly see the cancer markers. She calls them multi-dots. And she can make these multi-dots into lots of different colors, so that doctors could label multiple cancer markers all at the same time. Well, shortly after Jessica started working on this project in breast cancer, something unexpected happened to her. She became a cancer patient. Now, this is an unexpected turn in her story, and no one can explain it better than Jessica herself. Here she is last year, sharing her story with an audience here at the Museum of Science.
I went to the doctor, and I found out that I indeed had breast cancer at the age of 35. And it was a very interesting experience to be a researcher who understood cancer fairly deeply, but then become a patient. During my treatment, I had the full treatment. I had a mastectomy of my right side with reconstruction. I had chemotherapy and radiation. And it's now been 5 years since my last treatment. So hopefully everything's okay. Knock on wood. Thank you.
Well, it's hard to believe. There she was, working in breast cancer research, and she became a patient. Imagine how difficult it would be to overcome something like that. Well, cancer did not slow Jessica down. In fact, it inspired her. Now, before Jessica's diagnosis, she used to follow the same type of process that most scientists and engineers do. It starts out with a big idea that she'd like to test. To carry out the necessary experiments, Jessica needs to apply for funding, because science is expensive. If she gets funding, she can go ahead and carry out her experiments. If the experiments go well, she and her team will publish their results in an academic journal. Then they celebrate and move on to the next big idea. But after gaining perspective as a cancer patient, Jessica wanted to make sure that her research was having a real impact by actually getting it into the hands of doctors. And to do that, she switched to something called translational science. Now, the process starts out the same way, but there's a couple of extra steps. Now, before Jessica publishes the results of her experiments, she'll apply for a patent to protect her ideas. Then she can go ahead and publish. But that's no longer the last step in the process. Now, Jessica will apply for another round of funding so that she can scale up the production of her multidots so that they can make enough to actually test on real biopsies. And if the testing goes well, her research is translated out of the lab and into the hands of doctors, where it can have a real impact on cancer patients' lives. Jessica already had a way to make her multidots. Now, she needed to figure out a way to make a lot more of them. But in order to scale up for commercial production, you need to be well-versed in the business world. But Jessica is an engineer, not an entrepreneur. So she became one. Jessica participated in a National Science Foundation program called iCorps, which trains scientists and engineers to function in the business world. Then, she went ahead and started her very own company called Core Quantum Technologies. She hired Christy Melnick as her CEO and brought in some other fellow researchers and business partners to their startup space in Columbus, Ohio. Well, now that Jessica had her very own company, she was ready to start figuring out a way to scale up the production of her multidots so that she could get them to clinics around the country. But it wasn't going to be easy. You see, Jessica had been using a process called probe sonication to make her multidots, which is basically a blender. But this blender only makes tiny batches of multidots, and Jessica's trying to scale up her production so she can actually use these on biopsies. Well, for just one biopsy, Jessica would need 10 milligrams of her multidots, but that blender makes just 0.1 milligrams per batch. So that means that Jessica would need to make 100 batches for just one biopsy. So Jessica thought, well, maybe she could get a really big blender and put a lot of ingredients in it, and maybe she could hire a graduate student to make batch after batch in the big blender for hours on end. Would you apply for that job? I don't know if I would, but not everything in science works out the way we want it to the first time, and making bigger batches wasn't working. Instead of getting multidots, they got this, goop. Jessica needed a way to go from a blender to a high-speed, continuous assembly line for her multidots, like an automotive assembly line, but at the nanoscale. So she teamed up with two of her colleagues at The Ohio State University, Dr. Barbara Wislusel, who is also a chemical engineer, and Dr. Lisa Hall, who is a computer scientist and a molecular modeler. So Barbara can make a lot of tiny particles, like Jessica's multidots, really fast, using a process called electrospray. Now, it might be a little hard to see in this video because it's such a fine spray, but this electrospray machine uses a high voltage to continuously spit out tons and tons of these multidot particles really fast. It was the high-speed assembly line that Jessica was looking for. But in order to convince the Food and Drug Administration to approve their product, they need to make sure that all of those multidots come out looking and acting the exact same way every time. And that's where Lisa comes in. Lisa uses 3D computer simulations to figure out how those micelle capsules will form around the quantum dots under different conditions. Then they can use Lisa's models to adjust the electrospray process to make sure that all of the multidots come out exactly the same. Well, after a lot of hard work and determination, Jessica and her team were able to develop a fast, efficient, and reliable method to scale up the production of their multidots. And now, they were ready to look for funding and approval for commercial use. But first, Jessica had to show that her method for identifying cancer markers was better than the old method, using that brown stain over and over again and only testing for one marker at a time. Well, now, thanks to Jessica and her team, this is how doctors may be able to look at cancer markers in the future. This is an image from Jessica's experimental results. In this box, you might see some blue color, which is just a stain for DNA, so it's showing you where the cells are. but the other colors are actually the glow from different colored quantum dots labeling different cancer markers all in one single image. To take a closer look, we can split up the image so you can see that Jessica used a red quantum dot to label the normal cells, a green quantum dot to label one cancer marker, and a white quantum dot to label another cancer marker. These images confirmed that Jessica had achieved her goal of labeling multiple cancer markers all in one single picture, something that will have a huge impact on doctors' abilities to speed up the diagnosis process and get the most effective treatments to their patients in the shortest amount of time, saving time and saving lives. Jessica and her team have begun testing their multi-dots, and the results are promising. In fact, they've shown that their quantum dot technique is 2 times more effective than some similar techniques that are out there for testing for cancer markers. And they hope to have a new kit available for diagnosing in leukemia, commercially available by the end of this year. So stay tuned. Inspired by her own success fighting cancer, Jessica plans to continue translating her research from the lab to healthcare to benefit all of us. So let's give her a round of applause.